so I included that in this section here. For this talk, resource allocation is important when you have an immune response. One of the things we see in the fly, and I think this is certainly true in other organisms, is that people will show you microarrays of what happens in the fly, and they look at all of these antimicrobial peptides. But the thing that's really happening is metabolism has been completely rewritten in these sick animals. And so resource allocation is important. I want to take a second to talk about the point of view. So what I showed you here was really host-centric, where I showed you this chart where you have increased tolerance and decreased tolerance depending on what happens here. So Tom Little and Andrea published this opinion piece in PLOS Pathogens last year that made me have to redraw this. And I think you can. I think this works. That if you just change the axes and say, look at it from the parasite point of view, you'd have this thing, but it would be increased virulence down here and decreased virulence up here. You talk about the growth of the parasite. It's the same sort of movements would tell you what's happening if you're just looking at it from a parasite-centric point of view. And it causes some problems for me because I always think about things in a host-centric point of view. And I read a review on tolerance from an aphid guy that helped me out. And I think one way of discussing another tolerance mechanism is the following: that you can have bribery, blackmail, or gift exchange between the host and a microbe that could change things. So for example, with aphids, produce honeydew that they give to ants so that the ants eat the honeydew instead of eating the ants. Okay, you could consider that from the aphid's point of view as bribery. It's changing the behavior of the ant in order to prevent a fitness effect on the aphid. You could consider it from the other side round that it's blackmail. The ant is saying, give me this honeydew or I'm going to eat you. Or you could look at it from a purely mutualistic point of view and think of it as a friendly gift exchange. And depending on your point of view, you're going to come up with different ways of describing things. Okay. So that was to put some things in context for what sort of mechanisms we expect to be able to find. So I work in a medical school, and I'm always trying to tell people, so it's true that the immunologists understand immunopathology, but the first problem is when we start talking about tolerance, their brain freezes because they think you're talking about what they call immunological tolerance, and they can barely get that there's LPS tolerance too. But if you talk about other types of tolerance, it causes trouble. Okay. The other problem isn't just the word; it's that tolerance doesn't really help you diagnose people or help you change health outcomes in the short term at least. So imagine these were people who'd been through a malaria infection. A new sick guy comes into your office, and they have this point here. What are you going to do? So there are a couple of problems. One problem is you can put this point on the screen. This person has had to hit their maximum parasite density and their minimum red blood cell level. They're done. They're either going to die at that point, and it's too late to do anything about it, or they're on the road back to recovery, so it doesn't really help you. You can use earlier indicators. But the other problem is that tolerance is a population effect, and it really only makes sense when you're drawing a line through here. So what I don't know when this new person comes in, are they have they just fallen off this line? They're really part of this population. Or should I be drawing a new line through them? So I know, and the way I justify working on this is that with populations I can talk about how I can change the slope of the curve and play with it. But I started thinking about what can I learn about this individual and the shape of this individual's curve. And I want to go back to this animated guy that I showed you because I think we have some data that can help us here. So again, how can we summarize infections? So we'll just start from here. Microbes grow, he gets sick, he gets better again. Are there other ways we can summarize it, maybe using more of the data that we had to begin with? So we draw everything as a timeline because it's simpler to do the math when we do this, but it takes away some of the connections that we really know are involved in the process. So we draw this health timeline and this microbe timeline, but that's not really the way we think about things. What we think is that the microbes are driving this change in health. And so another way of looking at this would be to plot the data in phase space. So what I'm going to do here is show a movie where I go and I plot health by microbe number as we walk across time for the whole thing. So it goes like this. First you have this flat section, then the health gets worse, and then the health gets better. Okay? And what this shows is that there are three different portions of the curve. In the first portion of the curve, you've got health not changing as microbe number increases. You've got a second section of the curve where the health crashes while microbes are being cleared. And then you have a third section where the animal recovers, where the microbes are being cleared and health gets restored to normal. And this is actually the section that I want to spend the next couple of years working on, because typically in the lab at least, we can't look at recovery. So the way I do experiments is I take the animal and make it sick as hell, and then I measure mean time to death. So I'm only measuring how sick the animal gets and how fast it gets. It's like this. It's like I'm telling doctors in the emergency room that I can tell them how sick a patient is going to be when they show up in the ER. And maybe I can come up with ways of preventing that patient from being as sick when they show up in the ER, but I'm really not giving them any way of helping the patient get better. So I want to figure out this. Okay. So it's tough to see recovery in the published data. You can do it. It's actually even tougher because you notice in this data, granted they published it 10 years ago and they weren't thinking about this, but they don't even have the same timeline on the red blood cell count by parasite density curves. So even if you stretch it out, it's not so easy to make the correlations between when parasites are going down and when health is getting better. All the data that you need are there. It's just not easy to see. So instead of using this pretend data in the phase plot that I showed you, what happens when you look at the real thing? So what I've done here is I've plotted these 58 mice, and I'll show you the movie in a second. The red ones are going to get sick and die. The green ones are going to get really sick, but they'll recover. And the blue ones won't get very sick at all. So it looks like this. This is what happens. So, and I'll let this run a couple of times. So you see a couple of things immediately. The first thing is it's not a hairball. So this could have been just a mess, and there would be no recoverable information from it. But it's pretty regular. So the animals that die trace this outer ring over here, and they end up dying in this space, which is an interesting space because the parasite density is actually decreasing. They just can't turn the corner and get better. Presumably the reason is that when these guys go through one more round of replication, they've got no red blood cells left. Okay? So the animals die. There's sort of a Bermuda Triangle that if you enter, you're not going to recover. Another thing to notice is that the velocity that they move through this curve is important. So when it goes around the next time, even though there's some red ones that are coming up late, you can notice them because they move really quickly. And they're tracing the same path that all the animals take at the beginning, but the velocity is important. So it's also going to be important when drawing these curves, not just to measure position, but to measure the first and second derivatives of these, because I think that when you pay attention to that, when you see changes in the velocity of the animals moving through this curve, that'll tell you when different things are turning on. Okay. So when you think about that, so these are real data. This is one type of curve. You can extend it by thinking about the infections that we know about already. And I think that there are just a limited number of archetypical curves you can use to describe most post-microbe infections. So I'll walk you through this. Here's the one I showed you here, an infection, which is what we want, where you get sick and you recover. But there are other situations. Curve number 2 would be an example of something like encephalitis, where you end up with West Nile virus infection that causes permanent damage. You clear the virus, but you never recover completely. Curve 3 would be something like reactive arthritis, where you end up clearing the infection, but you end up with a permanent inflammatory disease. 3 is where I put tuberculosis, where you end up with a little Laca-Volterra-like curve, where these things cycle forever. You don't recover. 5 would be something like septic shock, where you can almost get the animal better, but you don't quite recover. And 6 is an example of, I don't know, flesh-eating bacteria that turns into compost. You don't ever recover it. It's turning into bacteria. And the thing I like about this is you can also use it to discuss mutualists. And I think that if we need to talk about host-microbe interactions, they always have to be included. So curve 7 would be something like a probiotic. You eat some yogurt, you're happy while it's going through you, but you poop it out and you're all back to your original starting state. 8 would be something like a live vaccine, where it changes your state forever. And 9 would be something like a mutualist that colonizes you and changes the way you're going to be forever as well. So, yeah, so a couple of points as I start to finish up. What I find really interesting about this, and what surprised me when I saw actual data, is that the path you trace through health by microbe space, what I hoped would happen, what I thought it was doing when I started this, is I'd be able to trace tolerance curves for individuals. I thought they'd get sick, and then they'd get better, and they'd come back along the same curve. But that's not what happens. And the curve you trace as you're getting sick is not the same curve that you trace when you're coming back. And so when we want to understand how you recover from infections, I think we have to be able to separate it and study it differently, because you're moving through a different space. So, can these curves tell us anything? Can you look at a curve and know something about it from looking at the curve? I think there's some things you can tell. If you start with a curve of a resilient system like this, if you look at bifurcations, I think you can make guesses about what's wrong with the system and how, why you're getting that bifurcation. So for example, if you're breaking off in this direction here, it's likely you've got a problem in clearance. Breaking off in this section here would indicate that you've got a problem in clearance and pathogenesis. And if you break off here, it would be a problem in recovery. So what I'm trying to do here is come up with, one way to do this would be to just come up with a set of differential equations that you could use to describe this, and then you can play with the parameters and see what the curves look like. And I've tried that. The problem is I've got so little data, I can make anything happen, and I can make anything happen in multiple ways. So what I'm trying to do is think more about the data I'll get, where I can get a curve, and then I can think about how the shape of the curve can change, and figure it ahead of time what's going to happen when I see these different types of curves. Okay. And then the last point I want to bring up had to do with health. So the data set here had monitored both red blood cells, weight, and parasite levels. So you can plot red blood cells versus parasite, weight versus parasite, or weight versus red blood cells. So I'm kind of happy plotting everything against everything else. And I don't want to stop there. What I'd like to do is do a microarray over the 21 days of this infection, and then plot, say, every of the 5,000 genes that are varying against every other 5,000 genes that are varying, and ask, what sort of curves do you get? Do you get curves that are telling you something? Now, all the data is there when we normally look at a timeline, but we don't look at it in ways like this. And I think that there's just a small number of correlations that you can get out of this. And I think that we'll be able to look at the, say, 5,000 by 5,000 varying genes and ask, do we get positive or negative correlations? Do we get no correlations where just one gene or the other one works? Or do we get these circles? And what you can do is decide, I'm interested in a certain sort of shape. So you can go looking for things that give you circles, and then find the genes that correlate with each other in this circular way.